Hello everybody, this is a presentation for the COIL program for October 24th, and me, Alejandra Carreira, will be presenting. So I'm going to present this paper from Nature Communications. It's called "The melanocortin action is biased toward projection from weight loss in mice." And we're going to start with a little bit of background. So for this paper in particularly, we're going to talk about the hypothalamus. The hypothalamus is this region of the brain that regulates many functions, and one of them is the energy homeostasis, and therefore like a body weight, increase of body weight, food intake, and energy expenditure. So in the hypothalamus, we're going to focus on two nucleus, the arcuate nucleus and the paraventricular nucleus. In the arcuate nucleus, we have, among other type of neuron populations, we have the agoutin-related protein neurons and the POMC neurons. And in the paraventricular nucleus, we have, among other neurons, the MC4R neurons. These are the melanocortin 4 receptor neurons, and we're going to focus on this. So the arcuate nucleus in general controls food intake and metabolism, among other functions, and the paraventricular nucleus is also related to these functions, but it's also relevant to point out that it constitutes one of the principal motor output systems on the hypothalamus. So let's go back a little bit. When we are on a fast state, there is a secretion of leptin from the adipocytes, and that activates agoutin-related proteins. This in turn inhibits the MC4 neurons, and that causes an increase in food intake. When we are in a fed state, the pancreas produces insulin, which activates the POMC neurons, and therefore these secrete alpha MSH that activates the MC4R neurons, and that produces a reduction in food intake and increase on energy expenditure. So this is known as bidirectional fashion of this pathway because one of the fashions reduce food intake and the other one increase food intake. But there are some evidence that suggests otherwise, and we're going to take a look at it as well. So first, one of the evidence that suggests otherwise is that loss of function of numerous mutations in POMC and MC4 and other genes related to the melanocortin pathway are associated with body weight imbalance, as it could happen here. As you can see here, as that happens when there is an MC4 absolute insufficiency, there is an increase on food intake. Then second, while it was expected that MC4 agonists, such as setmelanotide, could be leveraged as an anti-obesity drug, they only work for patients with obesity-causing mutations in the melanocortin pathway. And third, there are cases of obesity development associated with hyperleptinemia. So it's questionable whether this pathway can indeed promote weight loss, and this is the hypothesis of this research. So the experimental setup. In general, they're going to use this pattern, that is that they create several mouse models with chronic loss or gain of function in POMC or MC4 neurons. Then they will attempt to induce obesity and to reduce body weight or protect against obesity. And then they will create these models through gain of function or inhibition of the melanocortin pathway. So let's start with the first results. Chronic inhibition of POMC neurons causes obesity. So they stereotaxically inject a conditional adenovirus that contains KIR2-1 in POMC-3 mice. So this is a conditional vector that requires from CRE that will only be expressed on POMC neurons. So this plasmid contains diatomato and KIR2-1. KIR2-1 is a potassium channel used to inhibit neurons, and as overexpression of this channel will hyperpolarize cells. So first, they inject one side of the arcuate nucleus to verify that the plasmid containing KIR2-1 can reduce neuronal activity. After, to study the phenotype, they will inject in both sides of the arcuate nucleus. They will inject the vector with the KIR2-1, and then for the control group, they will inject only mCherry. This is a way to select the POMC neurons because the POMC neurons are not fluorescent per se. So by injecting with mCherry is a way to select them on FACS. To make sure POMC neurons were knocked out, they performed different experiments. So for example, they check for POMC peptide expression, such as beta endorphin, as is known to be sensitive to neuron activity levels. The immunostain for beta endorphin, which shows an obvious reduction of expression in the injected site. They also perform electrophysiological recordings on POMC neurons, and they exhibit a more hyperpolarized resting membrane. Then, for the in vivo part, after 10 weeks of chow diet, they found an obesity phenotype, expressed on body weight, and mainly caused by hyperphagia, which we can see in the food intake. In other words, they get fatter because they eat more. Second result, chronic activation of POMC neurons does not reduce body weight. So again, they do a serotoxic injection, in this case with an adenovirus that contains MAC-PAC in the POMC mice. NAC-PAC is a mutated version of the bacterial sodium channel that is broadly used to activate neurons. So, same as before, they first inject in one site to verify that the plasmid containing NAC-PAC GFP can increase neuronal activity, and after they start to study the phenotype, they inject in both sides. The control will have only a GFP. So, to confirm that the NAC-PAC expression causes chronic activation of POMC neurons, they measure CFOS expression. They observe much greater numbers of neurons with CFOS expression in the injected site compared to the control site, as well as prolonged action potentials and lower threshold, and therefore higher probability for action potential firing. They also found increased expressions levels of MSH, which is the figure 2D in F in here. Because, expression increased levels of alpha MSH and also of beta endorphin. So for the in vivo part, after 8 weeks post-injection, they found no body weight change and no difference between the NAC-PAC and the GFP. Then, to examine whether this chronic activation of POMC neurons could reverse any aspect of obesity, they fed a group of POMC mice with half a diet for 10 weeks, which induced obesity, of course. So this would be the case. And then these mice receive a bilateral NAC-PAC viral injection in the arcuate nucleus. After 8 weeks, no significant change on body weight and food intake were observed. Therefore, NAC-PAC expression failed to reverse obesity. Overexpression of alpha MSH in POMC neurons failed to reduce body weight. Given the known heterogeneity of POMC neurons, no change in body weight by chronic activation of POMC neurons might be due to non-melanocortin actions. To address this possibility, they generated a mouse model with selective overexpression of alpha MSH in POMC neurons, since an increased alpha MSH action has been considered as an increased melanocortin action. This will be the case. Same as before, they first inject in one side to verify that the plasmid works, and then they inject in both sides to see the in vivo. So they found an increase of alpha MSH, which is logical since they inject alpha MSH gene, but no difference in beta endorphin comparing to control. Thus, they achieve a selective overexpression of alpha MSH. For the in vivo experiment, they performed the bilateral injection and they monitor food intake after overnight fasting. Compared to the control group, alpha MSH injected mice exhibit a subtle but significant reduction in fasting refeeding. That would be this graph over here, suggesting that alpha MSH overexpression exerts an expected inhibitory action on feeding. But as we can see on figure 3E, the reduction appetite is only monitored in a short period. Okay, continuing with this part of the experiment. They want to examine the chronic effect of alpha MSH overexpression on body gain weight. So they monitor CHO diet feeding for 6 weeks and then high-fat diet feeding for another 7 weeks. Compared to control, mice with alpha MSH overexpression exhibit no significant differences in body weight fed either CHO or high-fat diet. To examine the potential effects on obesity, reversal by alpha MSH, they first fed POMC mice a high-fat diet to induce obesity. This will be this part of the experiment. To induce obesity. Then they delivered the adenovector, and again, they found no difference in body weight, food intake, or even fat mass. We can see these parameters in here. To further investigate the role of alpha MSH overexpression in reversing obesity induced by leptin deficiency, they delivered the adenovirus with alpha MSH to the arcuate nucleus of POMC OBOB mice. Since the OBOB phenotype cannot express leptin and therefore have an uncontrollable appetite. They monitored the body weight for 10 weeks and they observed no significant impact on obesity in OBOB, no significant impact in body weight in OBOB mice with an alpha MSH overexpression in the POMC neurons compared to the control. Fourth result, overexpression of beta endorphin fails to alter body weight. Beta endorphin represents the other well-studied cleavage peptide that, when released from POMC neurons, promotes feeding. Similarly, beta endorphin knockout in the POMC neuron inhibits feeding, which is independent of alpha MSH feeding inhibitory action. To examine the possibility that beta endorphin released from POMC neurons mediates body weight reducing effects, they deliver an adenovirus with beta endorphin to the arcuate nucleus of POMC mice. Same as before, they first inject in one side to test the vector and do a bilateral injection for the in vivo. So 4D. Because beta endorphin plays a role in analgesia, they also tested the analgesia response and found that beta endorphin might exhibit a significantly less sensitive response to pain induced by a hot plate. So they measure how long it takes to the mouse to remove the hand from a hot plate. For the in vivo experiment, they perform the bilateral injection and the control group have the GFP. Will be here. And they monitor the body weight for 6 weeks on normal diet, and then after 6 weeks, in half-fat diet. And again, they found no difference in body weight, fat mass, or food intake. POMC overexpression does not facilitate weight reduction. So POMC gene also gives rise to a number of additional peptides, which might also play a role in mediating body weight regulation. To test this possibility, they generate mice with overexpression of POMC by delivering an adenovirus that contains the mPOMC and GFP. In the unilateral injection, they measure abundant GFP, increases expression of alpha MSH and beta endorphin, which it can be seen here. They monitor the body weight for 6 weeks on normal CHO diet and subsequent 6 weeks in half-fat diet, and they found no difference in body weight, fat mass, or food intake, but they saw a significant increase in O2 consumption. Let's find it. Okay, in here, this difference is significant, which might explain the tendency of reduction body weight in POMC overexpressing mice. So as we can see, POMC peptides in the arcuate POMC neurons fail to reduce body weight in CHO or obesogenic high-fat diet conditions. Okay, so paraventricle, hypothalamus, MC4, 4R neurons are required but not sufficient for body weight regulation. To further verify the effect of melanocortin action on body weight regulation, they test the role of MC4R, which are known to primarily mediate melanocortin action on body weight. As the paraventricular hypothalamus is the major brain site that mediates MC4R action on body weight regulation, they target MC4R neurons in there. They created two models, one with a chronic activation that was caused by delivering a adenovirus with a knockback, and the other one with a chronic inactivation that was caused by delivering an adenovirus with KIR21. So in the chronic activation, they found, of course, an abundant C4s, while in the chronic inactivation, they found less C4s. When they measure then when they measure in the in vivo experiment for the chronic activation, they found no significant difference in body weight or in chow or high-fat diet. What would be this data in here? They also couldn't find change in feeding or energy expenditure, as well as locomotor activity. In the case of the chronic inactivation, which is the KIR21, they leverage the fast refeeding. They leverage the fast refeeding analysis, and they found less C4, suggesting inhibit neuronal activity. They also found increased body weight, feeding, reduction of O2 consumption, and reduced locomotor. This data is in supplementary data, that's why I didn't add it. Together, these results suggest that reduced activity levels of MC4R neurons lead to massive obesity, but increased activity levels of MC4R neurons do not alter body weight. Seventh result, MC4R overexpression in the paraventricular hypothalamus MC4R neurons fail to prevent obesity development. To overexpress MC4Rs in the paraventricular MC4R neurons, they injected an adenovirus with MC4R, and consequently they found more C4s, therefore more neuronal activity. It can be easy to see in here. Then they perform an alpha MSH treatment, which means to supply alpha MSH. And this also induced more C4s expression. When they perform electrophysiological recording experiments from brain slides, they found that alpha MSH induced more firing on MC4R neurons. As it's known that alpha MSH has an inhibitory appetite effect, they repeated the experiment in vivo, and they found that alpha MSH caused a significantly more inhibitory effect on fast refeeding in the MC4R mice. But without the overexpression of alpha MSH, MC4R overexpressed mice didn't display any body weight difference when fed either with chow or high-fat diet. The figure is this one. There is no difference in body weight, including no alterations in feeding, which is figure E and also F, or energy expenditure. MC4R neurons in the forms of increased neuron activity or overexpression of MC4R both failed to reduce body weight on chow or prevented obesity on high-fat diet. Then they created a mouse model with chronic activation, POMC neurons, and overexpression of MC4R to achieve that MC4R. To achieve that, they deliver an adenovirus with a knockback and adenovirus with the MC4R. So they monitor these mice, and they fed them for 8 weeks with chow diet and with other 8 weeks with high-fat diet. And again, they found no phenotypical differences. So the conclusions. These are my personal conclusions. And my conclusions of this paper are that orexigenic pathways are easy to promote, while anorexigenic pathways are difficult to promote. While this statement might sound simplistic, let's recap. As we have seen, different changes on the melanocortin pathway can lead to an increase of the appetite and therefore obesity. Inhibition of POMC neurons, loss of function mutations on MC4R and alpha MSH genes, or even chronic activation of agoutin-related proteins are sufficient to promote obesity. But producing a decline on appetite seems extremely difficult. They tried to suppress appetite by chronic inhibition of agoutin-related protein neurons, performing previous research, and also they tried by chronic activation of POMC neurons, or by increasing the POMC peptide expression such as alpha MSH or beta endorphin, and even by overexpressing MCR4 receptors. But nothing contributed to the reduction of body weight or appetite. Only the combination of overexpression of MCR4 and alpha MSH or an extra treatment of alpha MSH show a reduction of food intake, but this was only on the fast refeeding. So these results show that while acute appetite reduction is possible through pharmacological approaches, chronic inhibition of feeding can be compensated by adaptive changes in other feeding promoting pathways over time. For this, we can conclude that hypothalamic pathways that regulate appetite seem very robust, and in my personal opinion, they might have to be related with survivability. Another interesting conclusion from this paper is the melanocortin pathway, rather than having a bidirectional role, seems to have a more unidirectional role fashion, driving over obesity development, which again, I think it might have to do with survivability. So thank you very much, everybody, for listening. I hope to see you and have a nice discussion with you soon.